Hello students, welcome to EPG Pathshala. I am Dr. Seema Kalra from Indira Gandhi National Open University. Today we are going to discuss about the module Vaccinology 1, the basic concepts from the paper Molecular Therapeutics. You know that vaccines have proved to be a very effective tool or weapon for the mankind for the treatment of infectious diseases. In fact, after the development of vaccines, we have been able to control many of the deadly diseases like smallpox, yellow fever, which were taking toll of millions of people all over the world. So, today in this module, you will be able to learn answers to some of the questions like what are vaccines, how do they work, what do they contain, how did they develop, and are there any adverse effect of these vaccines? So let's address our first question. What are vaccines? The vaccines are the biological preparations which trigger our immune system to produce antibodies without subjecting the subject to the risk of the disease. So you got to remember that vaccines need to produce antibodies in our body for the effective protection process. Introduction of a vaccine into an individual is known as vaccination, and this term is sometimes used interchangeably with immunization also. And the purpose of vaccination is to induce immunity and protect against the diseases. Long before first vaccine was developed, there are reports that a practice known as variolation was done in China for prevention of smallpox as early as 1000 CE. It was also practiced in Africa and Turkey, and later on it spread to Europe and Americas. So this practice came from the observation that survivors became immune to smallpox disease. So physicians intentionally started infecting people with smallpox virus. This practice was of taking sample like pus from pustules or dried scab from the person having disease or who has survived the disease. So this material was either snipped through nose or injected into the healthy individual through cut in the skin. It was a very dangerous practice with inconsistent results and high mortality rate. In spite of that, it is quite popular as it gives little protection against the disease. This is a picture of Edward Jenner, who is considered as father of vaccinology. Jenner observed that milkmaids who were infected with cowpox were protected against this disease, which was a more deadly disease of humans. Cowpox virus is similar to that of smallpox, and when introduced in humans, it causes a mild disease but provides the immunity against smallpox for the lifetime. Based on his observations, he performed an experiment in 1796. He took a material from cowpox lesion of a milkmaid and inoculated into a young boy who was susceptible to smallpox. Then he waited for 2 months so that the child develops the infection and recovers from it. Once he recovered, he inoculated him with smallpox, and the child did not develop the smallpox. So it was the first experiment which showed the successful implementation of principle of modern day vaccinology. Although first vaccine by Edward Jenner was developed in 1796, it took almost 100 years before a new vaccine could be developed. So it was Louis Pasteur who actually gave the principle of vaccinology, that is isolate, inactivate and inject the causative agent. Based on this principle, he also developed various vaccines, and one of them was rabies vaccine in 1885. It was Louis Pasteur who also coined the term vaccine to commemorate the invention of smallpox vaccine by Edward Jenner, and the word was taken from a Latin word vacca, which means cow. By this time, various studies started coming up in which life cycles of bacteria and viruses were elaborated, and therefore the studies and developments in the field of vaccines were accelerated. In 1886, Daniel Elmer Salmon and Smith demonstrated that vaccines could be produced not only from live organisms but also from the killed disease-causing organisms. So this laid the foundation for development of inactivated vaccines. Then in 1890, it was Bering. He discovered the basis of diphtheria and tetanus. That is, these diseases are caused by the toxin released by these bacteria instead of the bacteria itself. And for this discovery, Bering also received the Nobel Prize. In 1920s, toxoid vaccines for diphtheria, tetanus, whooping cough and tuberculosis were developed, which were based on using the toxin released by these microorganisms to produce the immunity. In 1955, John Salk, he developed the first polio vaccine, which was an inactivated vaccine. 61, Albert Sabin developed oral polio vaccine, which came to known as Sabin vaccine. So in between, during this time period, World Health Organization, it resolved to eradicate smallpox from the world and launched an expanded program on immunization, that is EPI, in 1976. In this program, they distributed free vaccines to poor countries to expand the coverage of the vaccine among the population. And in 1980, smallpox was declared eradicated from the world. In the beginning of 19th century, various other developments took place in the development of vaccines, and 86 saw the first recombinant vaccine against hepatitis B virus. Quite recently, in 2008, Professor Harold Zur Hussain, he received a Nobel Prize for the discovery that cervical cancer is caused by human papillomavirus, therefore making possible the vaccine against cervical cancer. Now, let's try to understand how vaccines provide us the immunity. But before that, we shall discuss how our natural immune system works when we are exposed to any infectious agent like bacteria or virus. Human immune system is a complex network of various cells and organs. It is armed with variety of cells which are specialized to fight infections. So there are two components of the immunity, innate immunity and adaptive immunity. Whereas innate immunity is a non-specific immunity against disease-causing pathogens by preventing their access or entry into the cell or body through various physiological and anatomical barriers like skin, temperature, pH, mucosal membranes, or by destroying these cells through endocytosis or phagocytosis by cells such as macrophages, or through inflammatory responses if the pathogen damages the tissue. Second type of immunity is adaptive immunity, which is capable of specifically recognizing and selectively eliminating the foreign microorganisms or molecules. It displays specificity, diversity, memory, and differentiation between self and non-self. Now, not all the cells, they carry the certain markers on their surface, and all these markers, they are unique to that particular cell. So our immune system distinguish between a self and non-self based on these markers. Now these marker molecules on a microbe that identify it as foreign and stimulate the immune system to attack is known as antigen. Adaptive immunity plays an important role in identification of these antigens and subjecting them to elimination. To carry out its function, adaptive immunity has different type of cells, out of which antigen presenting cells and lymphocytes play an important role. Lymphocytes are further of two types, B cells and T cells, which when activated differentiate further into memory B cells and plasma B cells. Plasma B cells produce antibodies which are very specific to the antigen attacking the body. T cells, on the other hand, differentiate into further three types: cytotoxic T cells, helper T cells, and memory T cells. Cytotoxic T cells play a role in destroying the cells which are infected with the pathogens. Both memory B cells and T cells, they remember the antigen and therefore help in producing the immune response, which is much faster and much stronger when we encounter the antigen for the second time. Helper T cells are the cells which play a central role in the adaptive immunity. The immune response, the one which is mediated by T cells, is known as T cell immunity. Let's see how antigen presenting cells work. Antigen presenting cells are the specialized cells which can recognize the free antigen circulating in the body, and it alerts the immune system so that it becomes activated. An antigen presenting cell, it is basically a phagocytic cell which eats the bacteria, and a part of it, which is an antigen, it goes to the surface of this phagocytic cell. Now this phagocytic cell with the antigen and a specific protein known as MHC, that is major histocompatibility complex, acts as an antigen presenting cell which will present the antigen to helper T cell. These helper T cells, when they detect antigen presenting cells, they become activated. So this slide basically shows you how humoral and cell mediated immune responses are activated once there is an activated helper T cell. So an antigen which is circulating in the blood is engulfed by antigen presenting cell. This antigen presenting cell activates helper T cells, then activate both B cells and cytotoxic T cells. Activated B cell starts dividing into plasma cells, which will start producing lot of antibodies specific to the antigen, and it will also differentiate into memory B cells, which will remember the antigen for future encounters. Similarly, cytotoxic T cells will also differentiate into active cytotoxic T cells and memory cytotoxic T cells. So just like memory B cells, memory T cells will also remember the antigen for the second exposure. Now the antibodies which are produced by B cells and the cytotoxic T cells, the mode of action is different. While B cells, they are involved in destroying the extracellular pathogens in blood and lymph by binding to antigens and neutralizing these pathogens or making them target for phagocytic cells and complement proteins. On the other hand, cytotoxic T cells, they defend against intracellular pathogens and cancer by binding to and lysing the infected cells or the cancer cells. Here, another thing which would be worth mentioning is that when we are exposed to antigen for the first time, and the immune response which is developed to that is known as primary immune response, which is very slow because it takes time for body to produce antibodies specific to that antigen. However, when we are exposed to same antigen for the second time, the immune response is known as secondary immune response, which is much faster and much stronger than the primary immune response. So, this slide compares the action of a disease-causing organism and the vaccines which are inoculated to prevent the disease. So, vaccine is basically mimicking the antigen and activating the adaptive immunity. However, the difference is natural infection may result in one of the three outcomes: recovery with immune memory, or disability with immune memory, or death, whereas vaccine only results into immunity and the memory cells so as to improve our immunity and fight the infections. So basically, vaccines provide active immunity which is artificially acquired. Adaptive immunity is an acquired immunity which we get either through infection or from artificially induced immunogens. Naturally acquired adaptive immunity is further of two types: active and passive. Active immunity is the one when we produce antibodies in our own system, whereas passive immunity is when preformed antibodies are introduced into our bodies. Active immunity is when we are exposed to infections. Passive immunity we get when antibodies pass from mother to fetus via placenta or to infant via mother's milk. Similarly, for artificially acquired adaptive immunity, use of vaccines will be active immunity, and if there are any preformed antibodies which are introduced through serum or injection, they will be passive immunity. Let's see now what all a vaccine contains. So vaccine contains number of components which are required to protect its efficacy and potency. First is antigen. Antigen is, of course, it's the primary component of a vaccine. It's either a bacteria or a virus which causes the disease. However, in case of vaccines, these pathogens are inactivated so that they cannot cause a disease if they are live. And the process by which the virulence or the ability to cause a disease of a pathogen is decreased, it is known as attenuation. So basically, the vaccines contain either attenuated live pathogen or the killed pathogen. In addition to that, there are certain components of the pathogens which also produce immune response. For example, toxins in case of diphtheria, and then like some of the polysaccharides or other components on the surface of certain pathogens which cause immune response. So they can also be used as antigen for preparation of vaccine. Then another component is adjuvant. Adjuvants are the chemicals which themselves are not immunogenic, but when they are added to a vaccine, they enhance the immune response. Generally, aluminium salts have been used as adjuvants in many of the vaccines. Then antibiotics like gentamicin, neomycin, they are also added during the manufacture process of the vaccines. Once the manufacture process is complete, these antibiotics are removed, although some traces may still exist in the vaccines. Then the next component is stabilizer. Stabilizers are added to vaccines so as to maintain the potency and efficacy of the vaccines during the storage. Many organic and inorganic compounds are added as stabilizers. In addition to these components, we have trace components in the vaccines. Again, these components are also added during the manufacture process of the vaccines, but they still remain in traces, and one of the component which is commonly found in the vaccines is formaldehyde. Now, formaldehyde is otherwise toxic to humans, but the amount which is found in vaccine is in very minute amounts. So it is basically does not cause any harm to the humans. Then diluents, most of the vaccines, they come as liquids. So the components are in water or saline in the required composition. However, some of the vaccines which are dried and lyophilized, they are constituted just before the use by addition of these diluents. Now, in addition to all these components, there are preservatives which are added to the preparation of vaccines. The purpose is to prevent bacterial and fungal contamination of the vaccine preparation. It is especially useful in case of multi-dose vaccines where multiple doses are needed to be withdrawn from the same container. This table is showing some of the common preservatives like phenol, benzoathenium chloride, phenoxyethanol and thimerosal, which are used in various vaccines. Few years back, there was a controversy using thimerosal. It was linked to autism. So as a result, thimerosal is now not used in many of the vaccines, although these studies were found to be fake. Let us discuss about efficacy of vaccines. Impact of vaccines on public health is indisputable. In fact, their impact on reducing human mortality is second only to the provision of safe drinking water. Their importance in protecting the population from various diseases can be understood by the fact that many of the diseases which were prevalent earlier have been totally controlled and some have even been eradicated since the implementation of the vaccination programs. In India itself, the cases of tetanus, diphtheria, pertussis, and measles have decreased by at least 84% since the introduction of vaccines, and it has remained polio-free since 2011. So now this prevention and control of the diseases also boosts the economic development of a country because it saves the money being used for medical care. The ability of a vaccine to control or eliminate a disease depends upon two factors: effectiveness of the vaccine and the level of vaccination coverage achieved in a given population. Basically, vaccines, they work by a mechanism known as herd immunity, by which they can protect the entire population. Efficacy of a vaccine, it varies from 70 to 100%. Most efficient has the ability to induce the immune response in more than 95% of the population. But this alone is not enough. More of the population coverage is equally required. In this figure, you can see that when the population is not immunized and only few people are immunized, the disease can spread through the few people who are contagious. However, if you immunize more of a population, the number of cases will decrease, but the disease will still spread through the population. However, in the third case, where most of the population has been immunized with an effective vaccination, the spread of the contagious disease is controlled. So this phenomenon is known as herd immunity. Smallpox has been eradicated by this herd immunity. There is a report from WHO where the population coverage, which is shown in yellow line, was increased in 14 Eastern Mediterranean countries from 1980 to 1998, and it reduced the number of cases shown by yellow bars for measles significantly. The proportion of immune individuals in a population that will prevent the disease from spreading is known as herd immunity threshold. Higher is the threshold, more spreadable the disease is, contagious the disease is. So, but as the proportion of immune individuals in a population reaches herd immunity threshold, spread of a disease to non-immune population can be interrupted. Now, when the immunization campaigns are run, we want to know what is the impact of immunization. So it is measured in terms of vaccine efficiency or efficacy, which is basically defined as a reduction in the incidence of disease among those who have been vaccinated relative to the incidence of unvaccinated people. So you can also determine it by the formula which is shown, like it is a difference in the attack rate of the unvaccinated to vaccinated divided by attack rate in the unvaccinated population multiplied by 100. So basically it is expressed as percentage. Till now we have discussed how effective vaccines are in protecting the diseases. So you must be wondering, are there any adverse effect of these vaccines? Majority of the adverse effects which are associated with the vaccines are mild and transient, and these adverse effects are because vaccines, they induce our immune response, and people, they respond differently to different vaccines. So these mild and transient effects, they are like fever is there, rashes may be there, redness of the skin may be there at the site of injection. However, the more adverse effects like anaphylaxis are not known for any of the vaccines. So, students, let us summarize what we have learned in this module. Now, by now you must be understanding that vaccines, they play an important weapon for the mankind to fight the diseases, and it is because of these vaccines that many of the diseases like smallpox, whooping cough, and yellow fever, which used to take toll of millions of lives throughout the world, they have been controlled, and the quality of life and health has improved for the mankind. So basically, vaccines, they mimic the infectious agents and induce our immune system to produce antibodies which protect us against the diseases. Now, the vaccines, each vaccine is specific for a particular disease. So that means the vaccine which has been injected for mumps will not be effective against smallpox. Further, efficacies of the vaccines, they also vary, and even the most effective vaccine has been found to be able to induce immune response in more than 95% of the people. So the efficacy can vary from 70 to 100% of the population, and further that is because different people respond differently to the vaccines, and some may not even respond at all. However, if the vaccine which is preventing the disease and effectively controlling it, it can still be made effective to control the disease over the population if the population coverage with the effective vaccine is achieved. So that is called herd immunity. So if we can have the effective population injected to a higher percentage of population, we can definitely control the transmission of the disease across the population.